Hi everybody, I'm Dr. Tanner, and I'm going to talk to you a little bit about my research for INBRI this summer. My work focuses on developmental biology and genetics, and I'm really interested in the development of the intestinal barrier, as exciting as that sounds. Yes, I'm the poop guy. So I'm interested in this barrier formation. Remember, we have a single layer of epithelial cells that keep out the bad stuff in our intestine. So generally, we want all that stuff that we eat to come in through the center of the intestine, right, the lumen, and only the food comes across. And so this single layer of cells is that defense layer that keeps all that stuff there, and they're connected by these tight proteins here called tight junction proteins that help keep things from sneaking between the cells. Well, if we have a breakdown of that barrier, stuff comes through, and a lot of times you get a chronic immune reaction. And so you can see that down here. This is actually my intestine, and the reason I'm interested in this is because I have Crohn's disease, which is a result of a breakdown in that barrier. This is healthy intestine, and this is diseased intestine. So I'm interested in two different diseases, inflammatory bowel disease, as well as necrotizing enterocolitis, in which intestinal dies, and a lot of that happens because of the breakdown in this barrier in utero or in a preterm infant. So it's a very developmental related disease. And we look at this in the context of a gene called the multi-drug resistance gene, and we do this because polymorphisms in this gene or mutations in this gene have been linked to IBD and NEC. MDR itself encodes for a protein called P-glycoprotein or PGP, which is a small molecule pump, meaning it pumps out small molecules, namely drugs. We know it pumps out a lot of chemotherapeutics. And we do know that it is expressed in the intestine. MDR1 is the one that we're really interested in the context of humans. Now, my work focuses on two different models. We use C. elegans and cell culture. C. elegans is a small millimeter long nematode, so a little tiny worm that's been used heavily in developmental studies. They are all 969 cells, and we know where all of those 969 cells come from fertilization to adult. So they're a great developmental model. And then we use human cells in culture to study this as well. In the C. elegans, we're looking at the developmental process, and in human cells, we're looking at a wound healing process, with the idea being that if there's a wound, right, a little tear in the intestine, does it heal correctly to redevelop this barrier? And so we hypothesize that a change in intestinal function is actually due to PGP deficiency. So the previous data that we've collected with the worms, the C. elegans, is that if they're deficient in PGP, they develop slower. Their intestinal function is slower, the peristalsis is slower, and they show decreased expression of intestinal developmental genes and those tight junctional structure genes. And so we've done that through different ways, but here we can see a nice image of control, this LET413 protein that's being stained here on this C. elegans is one of the tight junctional proteins in C. elegans, and we can see that it is much less expressed down here in the PGP deficient worm, indicating that there's not as many of those tight junctional proteins, and maybe that barrier has been broken down. And so in cell culture, we actually found that the PGP deficient wounds healed faster. So this is a case where our original hypothesis and that they healed slower was false. And so we're trying to figure out why they heal faster, and if they do heal faster, do they make a good barrier once they've healed? And so changes in gene expression suggest it's an increase in proliferation, but we've not tested this. And so here you can just see one of the wounds that we've made, and then 2 days later, that wound is much smaller as those cells come together to heal that wound. So some of the things that I use in my lab are C. elegans, hair and candling, and cell culture, obviously, to take care of the models that we're using. We also use polymerase chain reaction and gel electrophoresis. We use immunohistochemistry or fluorescence microscopy, which is what you saw here, to look at expression of different proteins. Quantitative real-time PCR to measure gene expression. We measure cell proliferation and cell structure staining as well. So next steps with the worms is to create reporter strains to monitor the intestinal cells so that we can actually see the intestinal structure during development. We're going to look at the metabolic activity because we've seen that their intestines function slower, so are they actually able to process food as quickly and as well as control? And then look at gene expression during development. In the cell culture, we want to determine proliferation rates of control and PGP deficient cells, determine cell structure during the wound healing process, and look to see if those tight junctional proteins are forming correctly during the wound healing process. And then also look at the permeability after wound healing, right? So the idea there is, even though the wound is healed as far as cells coming together, are they still actually making that barrier? I do also have, just kind of as an aside, a virtual or remote project. So if for some reason the pandemic goes crazy, there is a kind of a backup plan, and that is looking at the insulin signaling pathway, the evolution of the insulin signaling pathway in Drosophila species. And so obviously there's a lot of similarities between humans and Drosophila over here and their proteins. And so we're interested in the evolutionary relationship in insulin in all of these different Drosophila species, using Drosophila melanogaster as our control. And we use things like NCBI's BLAST, Synteny analysis, Flybase's gene record finder, and the UCSB genome browser. But that's a summary of my research. If you're interested or want to learn more, please reach out. My email is st29@USC Upstate, and I'd love to talk to you. I find this really interesting, and I'm hoping to find students who find this interesting as well. Thank you.